Hi, science friends. Yesterday we talked about DNA replication, and today we are talking about the proofreading and repair mechanisms that happen following DNA replication. So, DNA replication, in the DNA, we have about 6 billion bases. That means 6 billion A's, T's, G's, and C's that have to be copied. And so that's a lot of letters. And the whole thing can be copied in just a few minutes. And so in a human, there's about 50 nucleotides added per second, okay? So they're reading A, T, G, and C and bringing in those nucleotides at a rate of 50 per second. So I want you to think about that for a minute, okay? When I type on the computer, I type about 40 words per minute, okay? Most people look at me and think that I'm a pretty fast typist, okay? But 50 nucleotides per second is equivalent to 3,000 per minute. So that's a lot, okay? That's a lot of letters that are being added in here. And so naturally, there are errors that happen in this process. So today we're going to talk about some things that happen and how those things are caught and corrected. And so one of the major players to work against these errors is an enzyme that we talked about yesterday, DNA polymerase. So we're just going to call it polymerase in general. We're not going to be so specific as to say what kind. Polymerase 1 is involved in this, but there's lots of polymerases, as we mentioned yesterday. So it proofreads the nucleotide and checks it up against the template as it's being added. So for example, when I'm typing, I know what I'm trying to say, and I know if I miss a letter. Sometimes without even seeing it, I can feel it underneath my finger, and I know that I hit the right thing or the wrong thing, and so I will backspace. Sometimes I'm typing from a textbook or typing from another document, and so I'll backspace without even knowing that I did it. And so that's kind of the action of polymerase. It's proofreading it, it knows it's made a mistake, and it will correct it as the strand is growing, okay? Now, there are errors that do occur in the bases. About 1 out of every 100,000 bases, you will have an error. You'll have a letter that gets, mistaken for something else. But most of those are going to be caught by polymerase, okay? Or another catch, which we'll talk about just a second, in mismatch repair. Only about 1 out of every 10 billion bases goes uncaught, okay? And remember, we have about 6 billion bases in our DNA molecule. So most of it does get, caught and fixed before it can go on and actually cause a problem. But after the polymerase does its action, the next is mismatch repair. So mismatch repair is like the second line of defense, kind of like spell check on a computer, right? Like, I know if I spell a word wrong, but if I don't, then spell check's going to catch it for me. So mismatch repair is like our spell check. And so it will remove and replace the incorrectly paired nucleotide, and then things that were overlooked by me or things that were overlooked by polymerase, okay? Now, even if it happens perfectly, right? Even if the DNA is copied perfectly, DNA molecules are constantly subjected to harmful materials, to cigarette smoke, to x-rays, to sun exposure, and they can undergo spontaneous changes because of these, okay? Now, sometimes mismatch repair will still go back and be able to identify those, but sometimes they don't. And, that can cause things like cancer or like genetic abnormalities that are passed down from, in the process, when babies are born, when the DNA is being passed from mom to baby, and in the process of making those gametes that go on to be sperm or egg, okay? So just because you have parents that, let's say, for example, you have two parents that are perfectly normal, they're not carriers for any type of disease, but they have a child with that disease. It doesn't mean that necessarily, most of the time the parents are carriers and they didn't know it, but also you can have spontaneous mutations that arose, and it wasn't necessarily because the parents were genetic carriers, okay? So I hope that makes sense. Now, another type of repair you have is excision repair. And so we see this a lot in damage from the sun. So what you see up here in this top picture is two thymines, okay? And if you notice there, those thymines, they're called thymine dimers because there's two of them. They are buckled. They are covalently bonded right here in the middle, and that causes them to change shape. That changes the shape of the DNA molecule. So this is something that can occur as a result of UV light. It's the exposure to the sun can cause those thymine dimers to join and change the shape of that DNA molecule. So there's other enzymes such as nuclease. Again, it's an ACE, so it's an enzyme that cuts the DNA. And so it recognizes this messed up sequence, it cuts it out, and then polymerase will go and fill it back in, just like that polymerase 3 did, like we talked about in replication yesterday. Okay, and then ligase, just like we talked about in replication yesterday, is the glue that will seal it back together. And so this is an example of nucleotide excision repair. Excision is to cut out, and so we're cutting out those nucleotides and repairing it with correct ones. And so that's something that you can that happens as a result of more like a spontaneous mutation. Okay. Now, another thing that happens, and this is kind of interesting, one other note on mutations. You can have mutations, but if they get corrected, they're technically not really considered a mutation. Only if they aren't corrected do they go on and be considered a mutation. Also, we'll talk about this more later, but mutations can be harmful or helpful. Generally, they're going to be harmful, not always, but they can be harmful or helpful. We think of them as harmful, but that's not necessarily the case. But all of them offer genetic variation, which natural selection can act on. Okay, so let's talk about that for a second. So, for example, we're right in the middle of this coronavirus pandemic, right? And so some of you all have heard of new variants of the coronavirus strand that started in other countries and have now made their way here. So what happened with coronavirus that caused those new variants? Okay, well, first of all, coronavirus is an RNA virus. And so that means that it's single-stranded, okay? It doesn't have the opposite bases across of it to keep it in check, right? In DNA, you have a double-stranded virus. And so when you have an A, it's going to pair opposite with a T. If you have a G, it's going to pair opposite with a C. And that pairing mechanism is a checking mechanism for it, okay? But in RNA, you don't have that pairing mechanism because it's just a single-stranded molecule. And so because you don't have that, then RNA viruses have generally more mutations, and those mutations can accumulate and they can change the structure of the proteins, okay? So right now, I got my coronavirus shot today. That was my mom just texting me a few minutes ago. You may see it on the screen to make sure I'm doing okay after that. But I got my COVID vaccine today, and the COVID vaccine specifically targets the spike proteins on the coronavirus. And it mimics the spike proteins, and so when your body, if you get infected with coronavirus, your body already recognizes those spike proteins, and it can mount an immune response against them. So ideally, if the coronavirus is mutated and changes, and we have these different variants, as long as the spike protein doesn't really change, then the vaccination is still good against the coronavirus, okay? Now, if the spike protein changes, then that's where we have a problem, because that's what the body recognizes to be able to fight the coronavirus. And so if that spike protein changes to a way that the body doesn't recognize it, then it doesn't recognize it as foreign to be able to put up that immune response. So for let's say that 90% of our population either A, has already had the coronavirus. It's not true. 90% is way too big. But A, has either already had the coronavirus, or B, has had the coronavirus vaccine, okay? So if there are a few people out there that are sick, that have coronavirus, normally they would transmit it to several people. In virology, they use a factor called R0, and R0 means how many people that one person affects. So the R0 of coronavirus is 3. So that means that if I have coronavirus, it's on average, I'm going to give it to 3 people, okay? So I would give it to 3 people if I'm one of the people that are infected. But if everybody has been vaccinated or already has had coronavirus, then I'm not going to be able, or 90% of them, like we said, then those people are not going to be able to get infected, okay? Now, so that's bad for coronavirus. That's good for humans, right? But let's say we have a different variant of coronavirus. We've had a mutation that resulted in genetic variation, and so we have a different variant of coronavirus that changed the spike protein, and so the people that already had the coronavirus and the people that have had the shot for the coronavirus, it doesn't really help against this new variant, okay? So then I can infect 3 people, or maybe even infect more people than that, and they can go on and infect more people than that. But the point is, that genetic variation helps the coronavirus to be able to spread, okay? Viruses and everything takes the path of least resistance, okay? The one that everybody has immunity for, it's not really going to spread. It'll eventually just kind of die out. But if there were a new one that nobody was immune to, then that's the one that was going to spread, okay? And so that's a source of genetic variation that we say natural selection will act on. Natural selection will select for the one that can divide and replicate easier. In our next unit, we'll go into evolution and we'll go into natural selection. But the goal of natural selection is to reproduce, okay? And so if it can reproduce, then it's the, survival of the fittest, and that's the one that's the fittest, okay? So that was in a little bit of an aside. Sorry for that side tangent, but I think it's important and really relevant to today and coronavirus, but also to changes in the DNA molecule, okay? Now, let's talk about one other thing with the DNA molecule. On the ends of the DNA molecule, you have something that's called telomeres. And so there's special nucleotide sequences. You don't have to know the nucleotide sequence, but it's TTAGG. And there are 100 to 1,000 repeats of TTAGG on the ends of the DNA strands. Now, the reason that that's important is because every time that the DNA strand replicates, those telomeres shorten, okay? So you've got this DNA strand, and the first bit of it is the telomere. The last bit of it is the telomere, okay? And every time the DNA replicates, you lose a little telomere, and you lose a little from this end. And then you lose a little, and then you lose a little more. And then you lose a little more, and then you lose a little more. And so over time, the DNA would get shorter. Now, these telomeres are non-coding sequences, okay? That's what this says here. They do not contain genes. They do not code for proteins. And so it's just kind of like gibberish, right? It's like if you're reading a book and you took out the table of context and the note from the author and the introduction and all those things. You still have the story. It's kind of fluff at the beginning and fluff at the end. That's kind of what a telomere is. But it protects the DNA from being cut down at the end, which naturally happens as the DNA molecule is replicated, okay? Now, you might ask, why does that naturally happen when the DNA molecule is replicated? Well, it's because of these lagging strands right here, okay? Remember, DNA has to be added in the 5' to 3' direction. So you can only add on to the 3' end. So this picture is we're just talking about this strand here. So we've got the 3' here and the 5' here. Here's our RNA primer. Well, when we go back to try to put DNA in the RNA primer, this is the 5' end. Down here is the 3' end. So you can't add DNA to that end because it's the 5' end. You can't do it. And so as a result, you have one strand that's a little bit longer than one another. And so that essentially is going to get degraded, okay? And then in further rounds of replication, it would get degraded further and further and further. Now, there is something, an enzyme called telomerase, that helps to lengthen out those telomeres. And this is specifically in germ cells. Now, what is a germ cell? A germ cell is something that's specific to reproductive cells. So this is only for meiosis. So remember, in meiosis, you start with one parent cell and it divides in meiosis 1 to 2 daughter cells, and they divide in meiosis 2 to 2 more daughter cells each for 4 daughter cells, okay? This beginning cell is called the germ cell. It divides a lot of times. The germ cells are where every round of meiosis comes from, okay? These are all germ cells. Oops, can't write, okay? In mitosis, it's not like that. All of the cells can reproduce, okay? Every skin cell on your body can give rise to new skin cells. Every liver cell on your body can give rise to new liver cells. But meiosis doesn't work like that. Sperm don't give rise to new sperm. You have the germ cells called spermatocytes that live in the testes that give rise to the sperm. And in females, you have the oocytes in the ovaries that give rise to the egg cells, okay? And so those are the germ cells. So these germ cells, because they undergo lots of rounds of cell division, it's really important that they remain at the full length, okay? We don't want those to end up being chopped up, because those are the ones that go on to create offspring, okay? And so for women, when they say, maybe you've heard people say before that you're born with all the eggs that you'll ever have. I used to hear that some, like, it was like something they would say to try to get you to like not lay in the tanning bed or whatever. They'd say you're born with all the eggs that you'll ever have, so don't like fry them all. That's what they're talking about, these germ cells. You're born with all the germ cells that you'll ever have. Those germ cells cause an egg to be released every month during your cycle, but you're born with all the germ cells that you ever have, and the eggs actually just come from that, okay? Same too with the men. You're born with all the germ cells that you'll ever have, but they continue to produce sperm continually. And so telomerase helps to lengthen those telomeres in germ cells so that you don't lose the genetic information, so that you don't lose the genes in the DNA, okay? Now, when you look at old people, if you looked at the genes of old people, they have undergone lots of rounds of replication, right? Because they're old. They continue to undergo rounds of replication. So their telomeres are going to be drastically shorter than younger individuals, okay? Now, telomerase works to correct the shortening of the telomeres, but it's insufficient to completely offset it, okay? So basically the telomeres are shortened faster than the telomerase can fix it, all right? Now, last thing I want to talk about, I know this lesson's gotten a little bit long, is chromatin. So chromatin, I'll come back to this picture in just a second. Remember, chromatin is what the DNA usually looks like in the cell. And so this is chromatin. It's just kind of a diffuse mass of genetic material. You only see it condense into chromosomes in when the cell's undergoing division in prophase and metaphase and all those. The rest of the time when the cell's not dividing, which if you remember that picture of the cell cycle, most of it was G1, S, G2, and then there was just like a small chunk that was actually mitosis. Most of the cell cycle is growth. But the chromatin is how the DNA usually exists in the cell. And so what happens with that chromatin? Chromatin winds up around these proteins called histones, okay? Now proteins, remember, are made up of amino acids, and these amino acids have a positive charge, and they attract to the negatively charged DNA molecule. DNA is negatively charged because it has a phosphate group. Maybe you remember biochemistry last year. Phosphate, if you pulled out that polyatomic ion sheet, it's right there. Phosphate is a -3 ion, And so the sugar and the phosphate in the backbone, the phosphate is the -3 ion. It has a negative charge. And so it attracts to this positively charged histone. And it coils into these structures called nucleosomes. And those continue to coil into bigger supercoils until they pack into a chromosome, which is what we see here. Okay? So if you look at DNA, chromatin is DNA plus proteins, the proteins being the histones, as we just said. If you look at DNA under the microscope, if you were to get a cell in cell division and look at a cell that's, in prophase, this is what it would look like. That's a cell in prophase, okay? And so it's not color-coded. They've made this color-coded for the picture. You can see here, though, that, there's maybe chromosome 5, and there's maybe chromosome, 11 or whatever. But they just kind of exist. They're, proportioned throughout the cell, and they have put them in a karyotype here. So when the DNA condenses into chromosomes, then that's what you see there, okay? And this is just another picture showing it, how the DNA is wrapping around these histones and then coiling up here. Okay? I think that's all for today. If you have any questions, let me know. You all have a worksheet, 17.1 review worksheet for today. And I will see you all tomorrow. Have a great day.